Kira, Kotu. Hello, everyone. It's my pleasure today to welcome Professor Matthew Lachniet, someone whose work has featured quite heavily in my literature review, so I'm very excited for this. Matthew is a full professor of geology at the University of Nevada, Las Vegas. He has also been the department chair for some time until the middle of last year. And his research focuses on studying past and present climate dynamics, mostly through the use of stable and light stable isotopes in geological archives, such as cave minerals. He's not going to talk about that today, I think. He's mostly going to talk about his work that he's been doing in New Zealand, and is currently doing, actually, as part of his Fulbright US scholarship that he's here for 6 months, I believe. Yeah. And yeah, I think that's all.
Okay, thanks, Todd.
Thanks for coming.
Yeah, thank you for having me. Great to be here. So I wanted to talk a little bit about some research that we have done and research that is building on that now that we're moving forward with some additional analyses. And I just wanted to at first acknowledge a lot of the collaborators that I've been working on with this. First, Drew Laurie is sponsoring me. He's at NIWA up in Auckland. So I'm affiliated with NIWA at the moment on the Fulbright scholarship that I have here. But this work really began with my colleagues at Otago, Chris and Christina, because they've been longtime friends, and they invited me down in 2016, which was on my last sabbatical to do some of this work here. And now I'm on my next sabbatical 7 years later, trying to improve on what we did the first time around. And then other collaborators, Seb has been really helpful. He came on one of the sampling tours before. Alice Hill at NIWA has done some sampling. Adam and a whole bunch of Otago grad students have been really helpful and kind of giving feedback on the project. I like to do these types of projects as collaborative as possible to hear other outside perspectives about how to do the sampling and the science as well. So I think it just kind of improves the process pretty significantly. So what I'm going to talk about are what we call isoscapes. Isoscapes are basically maps of stable isotope values. And I'm going to talk about the project here in New Zealand, which is focused on the South Island, and talk a little bit about how they're very useful for helping us understand climate dynamics in the modern perspective, but also as tools to help us interpret paleoclimate. Paleoclimate is what I mainly focus on myself with cave deposits. I'm not working on the caves here. There's already plenty of people working on caves here. But this type of stable isotope work is useful for understanding the signal that's preserved in cave deposits in terms of the oxygen isotopes in particular. So what I'm going to mainly talk about today is the paper that came out in 2021 that was based on the river isoscapes for the South Island and what we learned from that. And then I'll kind of lead into some of the broader implications of how we can use these isoscapes, and then look at the next steps, what we're actually working on now in 2024 to improve this, notably by adding the North Island, right? So we get a broader geographic coverage. So this is the paper that came out in Paleoceanography and Paleoclimatology, in case you're interested in kind of following up a little bit more with what we did. So the main question is, why do this? Why isoscapes? First, we have to ask, what is an isoscape? It's a map of stable isotopic compositions. So I'm going to go back to this first slide here. The colors that you see on the South Island are the inferred oxygen isotopic values of small rivers for the South Island. Right, so this is the isoscape that we produced here. And you can see the scale bar, blues are -4, that's a higher value. The reds are -11, -12, they're lower values. So what we're doing is building these types of maps to help us understand the climate dynamics. So I'm going to show you how we produce this map and what went into producing this map so that we can understand the ways that climate is controlling the spatial variations in the stable isotopes. Now, the reason we're interested in this is because paleoclimate records are using stable isotopes, either from cave deposits with speleothems, stalagmites, so they use the oxygen isotopes of carbonate. We use hydrogen isotopes in leaf waxes and in ocean sediment cores and in lake sediment cores. The Fiordlands National Park, where Chris Moya has been working a lot, they're using hydrogen isotopes there. And so one of the questions is, well, what's controlling those variations in the down core hydrogen isotopes or in the speleothem isotopes over time? Right, so the important thing to remember in all of this is that the stable isotopes of oxygen and hydrogen are strongly related to climate. So they're encoding climatic information. So from that, if we have a modern understanding, we can apply this to the past much better. We can also use this for stable isotope forensics. I'll give some examples of how we might use that for provenancing bird feathers or sheep's wool or human bones or teeth, for example. And also then relating this back to the climate dynamics. Right, so this is just a couple of examples of coring lake sediments or using cave deposits. So what we're primarily interested in doing here is using the spatial variations, essentially at one particular time, correlating the isotopic values to mean climate parameters. So we take a measurement of oxygen isotopes in the river waters and then correlate them against climate fields. So essentially we're substituting space for time. So we do very dense sampling at one particular time interval over a span of a few weeks, for example. And then we can relate that in terms of the spatial variation, which has climatic gradients associated with them. And then we can quantify the relationships between the stable isotopes and these climatic parameters. We can identify which climate process is the most controlling for the isotopic values. And that helps us develop predictive statistical models that allows us to create the isoscapes, these maps that anybody can then use to try to make a prediction of what the isotopic value at any spot in New Zealand might be if they were to sample a river. And then the final part is I'm going to talk about what we're doing now and updating this with more recent samples. Okay, before I get into the real science, I imagine that there are people here who haven't had a whole lot of experience with oxygen isotopes. So I just want to step back a little bit and talk about what oxygen isotopes are and the notation that we use. So we all know the water H2O, but it turns out there's many different isotopes in that water. And H216O is the more standard type, but we're also interested in H218O. So 18O is that rare stable isotope. So when we're looking at stable isotopes in water, we're actually looking at that ratio of how much 18O versus 16O is in that water sample relative to some sort of standard. So we use this notation called delta notation, where it just is looking at the variations of the 18O to 16O ratio in a water sample relative to standard mean ocean water. So the ocean has some value and the river water samples can either be higher than that or lower than that. So the delta values, when it's negative, it means you have less 18O than ocean water. When it's positive, it means you have more 18O. Mostly we're going to be in the negative territory, like -4 to -12, right? So that's where this delta notation comes from. And I'll be using this term delta 18O a lot, right? So it's that oxygen isotopic ratio in that particular water. And there's an equation for this that doesn't matter too much. That just to know that we're looking at those variations relative to the same ratios in ocean water. Now, we take this a little bit further in a subdiscipline that I like to call stable isotope climatology. So we all know climate, right? You measure the temperature, you measure relative humidity, you average it over a long period of time. Those are climate variables: temperature, relative humidity, wind speed, etc. You can look at different temperatures: maximum temperature, minimum temperature, median temperature, July temperature, etc. All those are different climate variables. I like to think of oxygen isotopes as also being a climate variable because it's so sensitive to climatic processes. It's just that it's not measured very much. But I like to think of oxygen isotopes as being a primary climate variable. And because we can make that connection, we can actually do climatology studies just with the stable isotopes. And the reason it works is that we have multiple isotope effects that control the oxygen isotopic values of rainfall and hence river water and groundwater, ice and ice cores, et cetera. So I'll just talk really briefly about a couple of those as a way of introduction to understand this, and then move on to how paleoclimate records like lake sediment cores or ice cores or speleothems are encoding this information in them and looking at those variations. So for example, this is a graph that shows what's something called the temperature effect, where we look at the delta 18O values of precipitation, which is on the y-axis, versus temperature. And you can see that there's roughly a linear relationship between mean annual air temperature and the oxygen isotopic values of rainfall. So this is known as the temperature effect, and it's the basis for a lot of paleoclimate studies that use ice cores. It starts to become a little bit nonlinear when you get into the higher mean annual air temperatures because you're in the tropics where temperature is less important. But at the mid latitudes to the high latitudes, temperature is a strong control on the oxygen isotopic values. This is just one example of the isotopic effects that links delta 18O for temperature. So you could see the utility of this kind of right away. Imagine you have an ice core that's 1,000 years old. You can then measure the delta 18O 1,000 years ago and have some estimate of temperature when there were no humans measuring temperature. So we can then use this relationship to make estimates of what temperature was like in the past. Another isotope effect, which is important, and I'll talk a little bit more in the context of New Zealand, is what we call the source effect. And that is that if you're at a particular location on the land surface, where that moisture source is coming from often has distinct isotopic variations. So if it's coming from the Pacific Ocean versus the Tasman Ocean versus the Coral Sea, for example, that moisture source variations will also be picked up at your particular location. So what you would do to measure this is you measure rainfall and the oxygen isotope values in rainfall, and you can see, okay, well, now we have a different source effect. If we have an ice core in one of these locations and it's varying between very negative values, maybe that's a continental source. If it's if they're higher values, then perhaps it's coming from a more low latitude moisture source. And then you can see these variations potentially over time. We have temperature effects, we have source effects. And then we also have seasonality effects. So this just shows a plot of monthly delta 18O values for several different sites. At the high latitudes in the bottom, this is 75 degrees north, so probably Greenland, I'm guessing. No, Northwest Territories and Canada. And so you see a large seasonal cycle with higher values during the summer and lower values during the winter. And then as you get more oceanic locations, that seasonality starts to flatten out a little bit. And then warmer locations like in the tropics, it's relatively flat as well. So when we're looking at paleoclimate records, they're always capturing some sort of the seasonality. So an ice core is going to be biased towards the winter because that's when most of the snow falls. Trees and might be pulling moisture from spring and summer, right? When they're growing and putting on their cells, right? So and then caves, you have to look at a cave by itself to know, is it getting mean annual or is it biased to one season or another? But this will tell us something about the oxygen isotopic value as it relates to seasons. Okay, so that just gives a very basic introduction of stable isotopes in the hydrologic cycle. Now I'm going to kind of narrow in with some more specific examples about what we did here in New Zealand and how we can use this to understand the climate dynamics of New Zealand itself. Okay, so what we did for this particular study is we spread out over the South Island and took river water samples. So you can see this is Chris Moy and Christina Rieselman from Otago. We, most of the times, just dropped a bucket off of bridges when feasible. And then other times we'll just walk down to the river's edge with a bottle and scoop out some water. You don't need that much, so we take anywhere from 20 milliliters to 60 milliliters of water. And then we do this for rivers all over, wide spatial variation, so we get all of the climatic gradients. Now, for this study, we were focusing on usually the smaller rivers. And the reason is those smaller rivers are more locally sourced, so that the climate at that sample site is more related to the local climate rather than some climate very far away. So some of the really big, iconic rivers in New Zealand, we didn't sample those. We just drove right over them and kept on going. The reason is they might be sourced from the Southern Alps 300 kilometers away, right? And so our sample location doesn't tell us anything about that local climate signal. It's sourced from somewhere far away. By focusing on the smaller rivers, we avoid that problem. We also avoid lakes or rivers with reservoirs because that water can evaporate, which then changes the isotopic composition and just complicates our lives. So we just focus on very small rivers that have little evaporation. Then we run the stable isotopes and we can correlate them against all kinds of different climatic parameters. And then do an uncertainty analysis, say, well, how good are our estimates? And then we can validate them by looking at previous river samples and estimate the values of those and see how well we can reproduce them. Okay, so what we do is we bring the waters back to the lab. This was an old way of doing it where we analyze the samples on a TCEA. Basically what it is you take your small little river sample, you heat it up to 1,400 degrees Celsius and it vaporizes, and then you can separate out the hydrogen gas and carbon monoxide gas, and then you can measure hydrogen isotopes and then oxygen isotopes. So this is kind of old way of doing it. I have a new instrument that's called a Picaro laser spec that's going to be installed, and I hope to take this TCEA and drop it off the roof of the building because it's horrible. It's so hard to work with. But basically, once you do that analysis, that's when you get your oxygen isotopic value for all these different rivers. Just some scenery photos. This was actually from this year. So on the upper left, this is Seb here risking his life over these railroad tracks to get a water sample. Myself here, I even got my 11-year-old daughter to collect samples for me, Chris and Christina. So it just kind of shows you the types of rivers that we're going to. I like to say that this is really a study of the streams, creeks, and burns of New Zealand, right? Because almost all of the sample names are streams, creeks, and burns. There's a couple rivers here and there, but rivers are usually reserved for those larger ones. So I'd say most of the time we're doing these hand samples, and that simplifies our life a little bit because when you use that bucket, you have to sterilize it after every sample so you don't add contamination or spread contamination. So we always try to get the hand samples whenever possible, which is usually possible. Maybe 80%, I estimate, of our recent samples are just from walking down to the river. So then you end up with, a pretty wide distribution of these different rivers, and you end up with your sample bottles here. So we've now, for the original study, we had 193 samples. For our current study, we're at sample number 300 something and continuing. So we're sampling all these all over the place. And I like to show this image, the Wairau River, because now we can also link these isotopic values to things like wine production, et cetera. This is where you get into the climatic forensics. So I bought this bottle of wine a couple weeks ago because I saw that it was one of my sample sites. You can see the little bottle there, says the Wairau River. Okay, so let's get into the actual science here. This is a map showing the digital elevation model. So just altitude for reference there. And then all of the blue dots are the river samples that we collected on the South Island. I think, we had 16 days and we just basically drove around the South Island nonstop, collecting river samples. You wake up at sunrise, you drive to a river, you collect it, and then you drive to the next river, you collect it. You do that until sunset, and then you sleep in a hotel, and then you keep on going until you've covered as much as you can. So on average, it was roughly 5,500 kilometers of driving. The cyan spots are precipitation isotope stations, which are useful for us being able to ascertain how well the isotopic values in the rivers are related to the rainfall. So you can see we have samples pretty much everywhere there are roads on the South Island, with the exception of Kaikoura, because right in our sampling campaign is when they had the big earthquake in 2016 and all the roads were closed. So that's our big gap. We have it in 2024. So we go to really colorful places like Roaring Swine Creek. And I love how the rivers almost always have these yellow signs with the names here. So I'm just showing you some of the isotope values. That was -7 per mil. The Fox Glacier outlet river was -9.7 per mil. The Pelorus River, which is up by Nelson, was -6.4 per mil. The Tasman River, -9.6. So that's just on the other side of the Fox Glacier. So they have essentially identical sample values. And it's because they're both being sourced from ice that's just flowing in different directions. And we even did the Spates Brewery spring tap just for fun, -6.4 per mil. That's down by Dunedin. And its value is similar to all the rivers around Dunedin as well, too. But it also does show you some implications. If you want to measure stable isotopes in beer, you can, right? And it'll tell you more or less the geographic origin of the water that was used to create that beer. Okay, so 193 river samples, 193 data points, making sense of it all. What does all of this mean? This is the real question, right? So you have this messy set of vials. How do we put it into some sort of order that we're understanding what's going on? And coincidentally, the picture on the right I took first, and then I accidentally knocked them all over, and I was like, oh, this is a teachable moment right here. So it's like playing dominoes. But how do we make sense of it all? What's controlling these isotopic variations? And then what can we do with that information? So the first thing we did is we just plotted them up. Again, this map is the digital elevation model, so you can kind of just ignore those colors. But each one of the circles here represents the stable isotopic value of the river at that particular location. And the color scale here is blue represents the higher values. So that would be -4 to -5 per mil. The red has the much more negative values, -12, -13. So what you can see is like all along the Tasman coast, you're mainly looking at blues, shades of blue. Once you start getting a little bit more away from the Tasman coast inland, they start to turn yellow and green until finally you get around central Otago, the Lakes District, where a lot of them values are red. So, and then you move closer to the Pacific coast and they start getting blue and green again. So you can start to see that there's actually a spatial variation in the isotopic values. You can only have that if there's a climatic control on the stable isotopic values. If it were just random noise, you wouldn't see any pattern at all. So we already have some indication that there's some clear spatial variation here that can help us understand what's controlling that. So then we look at the tiny dots here, the very small cyan, those are climate stations. And those are allowing us to look at the climate parameters over this area here. And then that's what we can use to build our regression models to find out what's controlling these values. Okay, so these are just our measured values. Nothing interpolated, no statistics here. It's just this is what we measured, this is what we observed. Another thing that we're always curious to look at here is whether these river waters have been affected by evaporation. I mentioned before, if you have lakes, they evaporate and that changes the oxygen isotopic variation. And then we don't want those values because it'll tell you something about evaporation at a lake that's not necessarily related to the climate. one way to assess that is by making a line plot between oxygen isotopes and hydrogen isotopes. It's called the water line. The GMWL is called the global meteoric water line, and that is the line that defines the oxygen and hydrogen isotopic values for rainfall across the entire globe. So what we did is we plotted all of our data against this global meteoric water line, and it fit almost precisely, which is telling us that evaporation is not controlling any of our river sample isotopic values. And we expected that, number one, because New Zealand happens to be a very wet location. Number two, we avoided rivers that were coming from lakes. There's just a couple of cases where we took lakes. Lake Alexandrina we measured because Chris was interested and it was very evaporative, as we just threw that sample out. This is important because it represents essentially equilibrium in our isotopic values. So we know that there's not secondary processes unrelated to climate that's controlling. Okay, so let's start talking about some of what we learned from this. The first thing that I wanted to point out is we see that there's a clear altitude effect. The higher the altitude, the more negative the values. So if you look at this bivariate plot, oxygen isotopes on the y-axis and then catchment altitude on the x-axis, you can see as you get higher altitudes, the delta 18O values are more negative. And you kind of expect that because it's colder at higher altitudes. But the curious thing that we saw is that there's a real distinction between the Tasman slope, which is in blue, and the Pacific slope, which is in red. So they both had very similar slopes of that relationship, but they're offset from each other. That tells us something really important. It tells us that the moisture sources from locations east of the Southern Alps is mostly coming from the Pacific Ocean. And sites west of the Southern Alps are mainly coming from the Tasman Sea. So that's now we're learning something about the climatic dynamics of the South Island here. And it's also telling us that temperature is a strong control. We already kind of know that from general principles, but this confirms it. Okay, so that's just one example of how you can do a correlation between a physiographic parameter, in this case altitude, which is strongly related to temperature, and delta 18O. Well, there's a lot of other climate parameters that could potentially be important. So kind of we generated this really large data set of all the different climatic parameters, and we estimated that for every sample location. So for example, you can look at annual mean temperature, mean annual rainfall, wind speed, vapor pressure, temperature, solar radiation, annual precipitation. The list goes on and on of different climatic parameters. And so we ended up assembling this matrix of 117 different climate variables. And some of those were very similar, like June maximum temperature, and then July maximum temperature, August maximum temperature, et cetera. So you end up with this very large matrix. But then we have this large matrix and we can ask ourselves, well, what are the climate controls on the oxygen isotopes? So this just shows some examples from this data set that's called the Bioclim data set that's based on all of these white dots here from the New Zealand climate stations. And it's in each one of these graphs, it's delta 18O plotted up against one of these different variables. So where it looks like a more linear fit, it's a stronger fit. When it just looks like a cloud of data, it's telling you that that parameter is not controlling the isotopic value at all. So there's a kind of a couple that stand out. The first one, the top left, is just delta 18O versus delta 18O, so ignore that. But the third one from the left on the top row, that's the mean diurnal range in temperature, right? That has a pretty linear slope. Minimum temperature of the coldest month has a pretty linear slope. The temperature annual range has a pretty linear slope. But then when you get into parameters like mean temperature of the warmest quarter, it's just a scatter shot. There's no relationship at all. So visually, you can start to see that, yes, some of these parameters are important for controlling the isotopes, and some of them are not. So this is a relatively small matrix of, let's say 1, 2, 23 different variables, but we actually had 117 different variables that we can make these correlations against. So this just shows the correlation matrix, every parameter, 177 correlated against the other 177, including delta 18. Right, so you generate this very large data set. That's the only reason I wanted to show this is that it allows us to get very precise with the climate parameters. And then you come up with a correlation coefficient and a statistical significance. So you can find out which ones are really important. So this is too complicated. If we want to make sense of this, we have to simplify this somehow. All right, so keeping it simple. What we want to do is we want to take those 117 different variables and then just pick out the important ones. We can do that ourselves based on expert opinion. Say, okay, well, here's the ones that I think are important. Or we can let a statistical model do it itself. So we kind of did both. And to illustrate this, I took just a subset of the data. It's basically this transect that goes from the Catlins through Dunedin up to kind of the central Tasman coast. And the nice thing about this transect shown in the inset map here is that it isolates latitude as one of those potential variables. And it just gives you a nice geographic representation. So this very bottom plot is the altitude of that transect, more or less going from the south to the north. And you can see, you cross, no, actually it's going from north to south. You cross the Southern Alps, you go over several other different ranges and then over to the Pacific Ocean. And then you see that there's pretty clear variations in temperature annual range on the top plot. Lowest values on the inland the temperature annual range, you have the greatest range of temperatures in inland Otago. The coasts have less range because they're buffered by the heat capacity of the ocean. This next graph in light blue is December potential evapotranspiration, which is much higher in central Otago. It's plotted with the reversed axis here. Minimum temperature in July. So winter temperature is much colder in the inland locations. And then in the dark blue are delta 18 values of those rivers. So you can see that there's a very clear spatial structure here. You start off with values on the Tasman coast of around -6 per mil. They decrease as you go over the Southern Alps. They're lowest in the lee of the Southern Alps, not on the Southern Alps, but in the lee of the Southern Alps. And then they start to increase again towards the Pacific Ocean. Right, so this is just showing you that there's a clear spatial variation here. So we could make a model, for example, that says, well, temperature annual range does a really good job of predicting the delta 18 values. The R value is 0.77, which is a good, strong correlation for climate science. Or we could say potential evapotranspiration in the summer in December is a really strong correlation. Or minimum temperature in July is a really strong correlation, et cetera. So you can start to visualize this, yes, that there are relationships between oxygen isotopes and these climatic parameters. We can also ask ourselves the question is, well, what months seem to be most strongly correlated to the oxygen isotope values? So we have these climate variables for every month, right? So we can say, is it winter? Or is it summer? Is it autumn? Is it spring? Which season seems to be most strongly correlated to the oxygen isotopic values? And it turns out that it's winter, right? When you look at the strongest correlations, they're usually associated with winter climate parameters. This is telling us that winter seems to be the most important season for explaining the isotopic values when we collected these samples, which actually happened to be in November of 2016. So it's kind of an early summer signal, but the winter is the climate parameters that are most relevant to those stable isotopes. So I'm not going to get into too much detail, but this plot is just showing you the correlation strength. So whenever you get into the winter months, June and July, that's when you tend to see the strongest correlation with any of these parameters. Okay, so that helps us now quantify some of this as well too. Like if we wanted to say what's the relationship between temperature in July and delta 18O, then we can put a number on that. And that turns out to be about 0.6 to 0.8 per mil per degree Celsius. Okay, but then what we did even further, we take this 177 variables. A lot of those variables are telling you the same thing. Like July temperature and June temperature are telling you essentially the same thing because they're correlated to each other. So we had to use a statistical model that basically says, okay, let's just pick the strongest controls or the strongest correlations, and let's pick 3 or 4 or 5 or whatever of them and combine them all together, making sure that the variables that we choose are themselves not correlated to each other. So for example, you don't want to have July minimum temperature and June minimum temperature in your final equation because they're basically showing you the same thing. So what this set of graphs does is it shows you our overall model on the far left. So we have measured values on the y-axis. That's what we actually measured for those 193 samples. And then the modeled values, you can see there's a very strong correlation. In fact, the R value, the strength of that correlation is 0.88. Again, for climate variables, that's considered a very strong correlation. But which are the main variables that went up to make this regression model? So the model kind of chooses these itself because it says, okay, I'm only going to choose the strongest set of variables that are all not related to each other. And so we just let the model choose here. And it shows temperature, minimum temperature of the coldest month, evapotranspiration in December, so in the summer, surface radiation, solar radiation, so how sunny that particular location is, and then latitude. And then you can come up with an equation that relates delta to know all those different parameters. So that gives us a way to predict. And it also gives us a way to predict potentially into the future when temperature of the coldest month might change due to global warming or something like that. So rather than just kriging our data or making a spatial map and contouring it, this gives us predictive value in case any of those variables change. So we just let the model do this. And I'm just going to go back for a second here. So these maps on the right actually show you those climate variables over space. So you can start off with these maps, and then it's just manipulating the maps in terms of the algebra. So we have a map of every one of these different climate variables. And so we can say, okay, we know based on our equation here, which are the ones we want to choose. Then we just do algebra on the maps, right? And we can then combine these all together to produce that final isoscape. Right, so this isoscape right here is based on these 4 variables. So we just combine them all together, and then it'll give us a predicted isoscape, for that set of data. And so you can see it fits with what we had originally done, which is showing the circles for each of our values in terms of the colors. Is that you have this swath of blue along the Tasman Sea. And it turns out that latitude is not that important. Fiordland has more or less the same values as Abel Tasman way up in the northwest. Right, so the rivers are pretty consistently giving that same signal along the Tasman Sea. And then the values start to decrease. They get more green and red when you hit the Southern Alps, the peaks of the Southern Alps. And then they go really negative in the Lakes District around Otago. And then once you get closer to the coast, they start to get more green and blue again. So this is showing us that the minimum values are really in Central Otago. And if you've ever been to Central Otago, there's a couple things that stand out to you in terms of the landscapes, in terms of the climate. It's one of the driest places in the South Island. It's one of the coldest places in winter because it's isolated from the oceans. These are related to continentality. We don't think of continentality as being a big issue in an island, but it turns out that if you're looking for the most continental location in the South Island of New Zealand is going to be right around central Otago. And that's also where we see the most negative delta H&O values. So we can kind of zoom out here a little bit and say, well, all of these different climate parameters in this model, those 4 different ones, they're all pretty much relating to continentality. And what it means essentially is that the most negative values are in the coldest areas in central Otago that are most distant from the moisture sources of the Tasman and the Pacific Ocean. So this map is publicly available. You can pull it into a GIS and click on any grid cell and it will tell you the estimated delta H&O value of a river if it were a small river basin from that location. Okay, so when we produced this, we submitted our paper. The reviewers were really critical of a lot of the things that we did. And they said, well, one of the things you're missing here is seasonality. Like these rivers are probably changing from winter to summer and you just have a spot sample, right? They can be affected by weather that's not related to the climate means, all kinds of different things. So we wanted to ask a bunch of questions about how representative our data were for explaining kind of these spatial patterns. In other words, is this map only useful for November of 2016 when we collected the samples? Or does it have some broader use as a climate variable, as a climate space? And without doing these tests, we don't really know. So the reviewers really pushed us hard on this and said, okay, well, you really need to assess this. Otherwise, we don't know if you're just looking at some random pattern that showed up once and might not ever show up again. So we did uncertainty analysis. In a couple of ways, there's a bunch of different ways you can do this, but one way is saying, okay, let's take 193 samples and let's run our regression model on 80% of the data and then estimate the remaining 20% and compare them. How well did it compare those samples that we left out? So that's called a jackknife approach. So we ran this model 1,000 times, each time leaving out a different 20% of the samples. And we can estimate how good our data is in explaining our own data. And it's pretty good. We're usually within 1 per mil, which is pretty good. But that doesn't tell us a whole lot because it's using our own data set to estimate our own data set. It's better if we have independent data. Actually, before I get to that, I'm just going to go back for a second. This maybe was a little bit out of place. So this map here basically shows our interpretation of what's happening, with the moisture sources here. The Southern Alps are blocking most of the Tasman moisture. And the other ranges are also blocking the Pacific Ocean easterlies. And that's why we get the most negative values in the middle of New Zealand. Okay, so to summarize that spatial variation, you get westerlies from the Tasman coming in, highest values, they get more and more negative as you get more distant from the moisture source, and the rain shadow, we call the isotopic rain shadow, and the same for the Pacific. Now, what did we learn kind of from this basic process here? We learned that winter temperature and continentality are really the most important controls, and that we have these really negative isotopic values in the lee of the Southern Alps. I'll also emphasize that we didn't sample many glacierized catchments. We tried to avoid the ones, that had glaciers in the headwater, because that could be very old water. We had a couple here and there, but for the most part, not. And then that also means that if you're working with isotopic proxy data, like in lakes or in caves, it should be reflecting winter temperature. This gets back a little bit more to testing our data against other independent sources. So there's this source called the Oxygen Isotopes and Precipitation Calculator, which is shown in this graph on the left. And that kind of makes an estimate for the whole Earth, all of the globe, and I just isolated New Zealand here. And so I said, okay, well, how well does this isoscape work for New Zealand? And it turns out it doesn't work very well because they didn't have much data to input into it. So just for one example, here's Lachniet Creek, this is near Wanaka. We measured a delta H&O value roughly minus 13 per mil. This OIPC map suggests it should be around minus 9 or minus 10 per mil. So it's not even close. Our Isoscape estimates it as minus 12.7, right? So using our data to interpret our data, it's very good, as you would expect. All right, so this map is not very useful. The OIPC map is not very useful for New Zealand. And the reason that's important, in my opinion, is a lot of people use this because they don't have their own data and say, well, we'll just use this OIPC map and make an estimate. But I'd say don't do it, not for New Zealand, because there's not enough data here. There's also a precipitation isotope map for New Zealand from Basden et al. I'm not sure if I'm pronouncing that right. If I'm not, sorry. And that's a physically based precipitation isoscape shown on the left here compared to ours. Now they reverse their color scale, but it basically shows the same thing where there's more negative values in central Otago. So we can, look at Lachniet Creek again, just that one example. It's hard to tell exactly from that precipitation map because the data aren't available for me to, click a grid cell. But it looks like it's around minus 10 to 11 per mil, and we measured minus 13. So again, it's significantly off. Another way we did this is using our data to kind of retrospectively predict values that were measured back in the 1970s and 1980s by Stewart et al. So this was a really great study where they published a couple hundred rivers on the North and the South Island. We said, hey, great, we sampled some of those same rivers. How well does our isoscape predict the values that were measured 30 years ago? If our isoscape is just kind of random weather noise, you would expect there would be no relationship at all. So we found 30 years, I don't remember how many rivers it was. It was roughly 30 or 40 different rivers that we both sampled, the Stewart et al. And us. And we've got more than 30 years, 35 years of time difference here. And it turns out that the mean difference between our isoscape and their values was plus or minus, or was 0.09 per mil difference. So essentially identical, plus or minus 0.68 per mil. So what this tells us is that this isoscape is really reflecting some kind of time mean. And it's useful for predicting values that were measured 30 years ago, right? So it's just more validation that the river values that we're measuring today are a real climate signal. There's no other way to explain this. Another thing that we did is we said, okay, well, presumably it's rainfall that's making the river water. So how well can our river isoscape predict the delta 18O values of rainfall? We're kind of inverting this process, right? Because usually it's the rain that makes the rivers. We're saying, if we know the rivers, how well can we predict the rainfall? And so we can do a measured versus predicted model. And again, we're within 1 per mil. When we look at either winter weighted average rainfall versus our isoscape, or just mean annual rainfall, we do a pretty good job. Within 1 per mil, we can predict what the rainfall value will be just from our river isoscape. Again, this is not possible unless that river isoscape is really giving us a climate signal rather than a weather signal or some kind of random signal. So I feel like we have some pretty good confidence in this approach, but there's still a lot of uncertainties. I'll get to that in a second. So let's just step back for a second and say, what did we learn from all this and why is it important? Well, we learned that there's a clear spatial variation on the South Island. We learned that oxygen isotope values are strongly correlated to continentality and winter temperature. The spatial variations are likely a robust and real climate signal, and they're reflecting the climatic processes that are producing the rainfall that's then flowing into the rivers, rainfall and snowfall. And then we can use these isoscapes in a lot of other disciplines. And this is where I think it becomes really interesting. So what are some of the broader impacts of this? So one of them is in terms of geology. There's a really important paper that was published in 1999 by Chamberlain et al. And what they did is they went in these sites in central Otago around Alexandria, and they looked at the hydrogen isotopic value of kaolinite clays. And they were able to show that over roughly 3 to 7 million years or so, the isotopic value of those clays had changed, and they interpreted it as the uplift of the Southern Alps. So when the Southern Alps rose, it changed the isotopic composition in this location. So they were able to show, using clay isotopes, that they can do something really powerful with that. And say, well, actually, we can estimate the delta value, the delta H&O values of clays. So this is simply my isoscape model run through an equation that converts them into the clay isotope values. So assuming that those clays are formed with the same source of the water that's in the river, you can now say, hey, if you find a clay, it's going to have its lowest values in central Otago and the highest values on the coast. So this is useful for geology, looking at uplift of the Southern Alps. And they happen to sample exactly the right spot because that's where the biggest isotopic difference is right where they sample. Another way we can use this is by looking at caves. So again, this is the river isotope map converted into the oxygen isotope value of calcites that would precipitate in a cave at equilibrium. So you have to know something about temperature as well too. So we took the temperature field, an equation that relates oxygen isotopes in water to calcite. So if you find a speleothem, a cave calcite, you don't know where it came from, you can start to use the oxygen isotopic values in this map to try to pinpoint where it might have come from. Right, so this is just one example of how you can use this. Another example is looking at organics, leaf waxes. So if you have plants growing along a river that are pulling in that river water, they die, the leaf waxes end up in a lake sediment core somewhere, well, then that's going to also be related to this. So this is a map of deuterium, not in leaf waxes, but deuterium, the hydrogen isotopes over New Zealand, which then you could convert into a leaf wax. Another impact is you can use bird feathers, hydrogen isotopes in bird feathers. So we all know the kiwi, right? And all these other things. We can figure out if you have a kiwi feather, you can estimate where it could have come from based on a bird feather map. So again, use the same isoscape, an equation that relates feather hydrogen isotopes to water hydrogen isotopes, and then boom, there you go. Right? So the kiwis coming from the Tasman coast are going to have higher values. The kiwis coming from the central Otago region are going to have lower values. You can start to see this is useful in bioforensics. You can do the same thing for sheep's wool. Make maps of the expected values of sheep's wool. You can do it for human hair. If you're dealing with archaeological context or, God forbid, murder victims, right? If you want to know where they were living when their hair was growing, that's going to be reflected in the stable isotopes of the hair or in the tooth carbonate in the skull, right? When you grow your teeth, it's equilibrated with the water source that you're using. So these are just all maps that you can produce here just by some relatively simple equations that have a lot of applicability in other fields. And then we can look at time series of paleoclimate records. So this is a speleothem composite that came out a couple of years ago. Adam is on this paper. And help us interpret, the oxygen isotopic values over time. And you can see that they're changing. You have this broad scale change over the last 12,000 years or so. So we can interpret the more negative values as being either colder or more winter moisture. The higher values as being a little bit warmer or less winter moisture based on what we know from the isoscapes and the correlations between delta H&O and temperature. Right, so that just gives some idea of the utility of this type of work. There's still a lot of unanswered questions, and that's where I get to kind of the ongoing work. So I've been here since January, driving around. So far, about 13,000 kilometers of driving with Seb and some other people sampling rivers. So this is what we've managed to sample just since January. We're pretty much done. But we're going to be going back to the South Island 2 weeks and doing one more kind of repeat sampling. So we're trying to do at least half of the samples repeated. So we have values, for example, in March and then again in May, and values that we did in April and again in June, just to see if there's any seasonal variation. And the idea is we're going to try to do some more sophisticated statistics that can also relate not just the sample location, isotope or climate parameters, but the drainage basin parameters. So that inset map shows you, it's kind of hard to see here, but it shows you the drainage basins above our sample sites. So from there, you can actually say, okay, what's the mean annual temperature for that entire drainage basin, not just where we sampled, right? So we can get a better spatial representation with this future data set. We're also going to be using the more up-to-date NIWA coverages for the climate here rather than the Bioclim data set, which is more global, lacked some of the local expertise. So this is really what's to come in this particular project. Once I get back to my lab in July, I'll start analyzing the second batch, and we'll move on to see what else we learn. There's also another difference is that we're sampling at the end of the summer and into the autumn compared to early summer from 2016. And we're repeating a lot of the same rivers. So we'll have kind of same rivers over time to see how much they vary. And one of the things I'll do is I'll take my isoscape and predict the 2024 samples, right? I'll do that before, and I'll see how good I am, see how good our data is for doing that prediction. Okay, so I'm pretty much wrapping up here. The conclusion I've already said several times, and looking to the future, that's just what we're doing now. So I think I'll just wrap it up there and say thank you for your attention. Any questions online or in the room? How do I view this question?
It would be go into the
Zoom workplace?
should be able to change. So you should be able to view the
Here's chat. Okay, Dudley BD. Go ahead, type your question if you haven't.
Can I just talk?
I'm either doing something wrong or I'm not seeing anything.
Yeah.
Okay, he's typing. Yeah, we're not hearing any audio here, so you just have to type a question.
Might just ask while we wait.
Okay.
That's okay.
Sure.
Maybe you explained it and I missed it. So with your multiple regression model, your different climate variables, some of the most important ones seem to have summary statistic reflects a slightly different season, depending on what summary statistic you're looking at for those variables.
Right.
So it was a December value. I can't remember exactly for what it was, and some other seasons in there. So how easy or how difficult is it to actually untangle the actual underlying mechanisms of those variables influencing the isotope value? What's the process governing, the isotope values that are reflected in these different climate variables?
Right. So the question was basically in our multiple regression model, we had a couple of winter proxies and then one summer proxy and then latitude. Right. So how do we untangle which of those is potentially the most important, or how it's controlled there, and how do we kind of deal with the seasonality? So the summer proxy was evapotranspiration, which is an indicator of continentality. So I would say in that case, we're really seeing that the central Otago region just has a more continental climate, and that's why the delta H values are lower. It's not that necessarily the summer evapotranspiration is controlling the value. It's that it's itself a stand-in for the sources of moisture that have to be coming from the oceans. But to go further is you can isolate single variables as well too. You could just say, well, what's the correlation between delta 18O and a single variable, and then you'll end up with that. The statistical model just gives you a little bit of extra better fitting when you combine multiple values at a time, which is why we went with that. And so I would conclude that it's almost primarily continentality that's really controlling this, rather than those individual factors in the summer. Okay, good question. Okay, so oh, awesome, Alice is here. Yeah. Okay, so so the question is, how can your sampling aid development of our precipitation model? Oh, this is Bruce. Awesome, glad you're here. We're having trouble replicating orographic effects and the east-west patterns across the southern Alps. Small streams seem potentially useful. Yeah, I would agree. Ideally, when we have a precipitation model, we'd like to have a very high density of sample sites, but that's very difficult to do because it's so time consuming to set up all these different precipitation sampling collections everywhere. And that was one of the motivations for doing our study, is we felt, well, it's difficult for us to constrain precipitation ourselves because it's so time consuming. Why don't we substitute space for time? And I think in this case, Bruce, that I think we should use your river model and the precipitation model and use our data just to see how well they compare. And then the idea would be to look at the residuals between the two and ask ourselves the question of whether or not there's something we can do to better interpolate the precipitation isoscape model where there aren't stations, I think. I wish we had audio so we could turn this into a conversation, but I don't think we have that. So anyways, I hope we can talk some more, Bruce, about this and get exactly at this question. So if you have maybe a follow-up question, go ahead and write it in there, or any observations of your own, and I'll move on to Alice's question. Yeah, so Alice is also involved with this in this project. Alice is at NIWA in Nelson, and she sampled some of these rivers as well too. So the elephant in the room with New Zealand is characterizing the influence of moisture source and direction. For example, the northwesterlies versus the southerlies, and they have different isotopic signatures. How to incorporate that into predictions, since it's not part of the large climate data sets that are typically used as predictors? Yeah, this is a really tough question. So basically the question is, if you have different moisture sources, how would you know it if you only have one or two samples from a particular location? Repeat sampling is what we really need here. And I think the approach that I would like to see is really doing the back trajectory analysis on event-based sampling. And I'm not sure to what extent that's been done in the precipitation data sets, but I think that would be the nice place to really move next for making this question and looking at the relative proportions of the different moisture sources. The rivers are going to be integrating over time. And so we can't do that very easily unless we're going out to the same rivers over time every couple of weeks or something. It's a very good question. I know that we're going to have more conversations. Yeah, and then Bruce is saying the river model is currently using the Bayesian precip model as input, and it does need some updating. I think we can do this multiple regression model on the Bayesian precipitation isotope data too, and maybe get some more variables in there, and maybe that will improve it for the NIWA river model. Good. Okay, I see a couple others. Say Thomas said there are a few time series data sets for the water isotopes monthly or a year or so. Did you manage to track down anything useful? We didn't look that in much detail. The only question we really had with that was how much seasonality there was in the river isotope data sets that we saw. But I didn't personally go into detail about the precipitation isotopes or other people's data sets, at least not yet. But we did see that in many cases, the seasonal cycle is relatively small for most of the places that we looked, but we really need to look in places like Central Otago more to see how much variation there is on a seasonal basis. And then Simon says, great talk, thank you. Interesting that you didn't include altitude. Would it be possible to include max altitude between site and coast in the model? So yeah, this is the thing, in the model, we can actually force it to put in the variables we want, or we can let the model choose it itself. We thought in this paper, we would let the model choose it itself so that there wasn't bias in there. But yes, we can do that. We can choose any model, any parameter that we want and come up with a different model. What happens if I do? Okay, try speaking. Simon. No, it doesn't work.
Okay. Hello? Can you hear me? Can't hear me?
Yeah, I think we just don't have audio.
I can hear you, Simon, but it doesn't sound like it's coming through.
Yeah, sorry, folks, we do not have audio.
We could be having our offline conversation here, it sounds like.
Yeah, I like that.
We could decide what to type in together.
Yeah.
I've got so many questions about this.
Okay, hey, we can hear you now.
Oh,
Yeah, we have a volume knob. So I guess maybe before we go to that, if anyone in the room wants to leave, feel free to leave. You don't need to stick around for this. Sometimes these can go on forever, but
it's gonna get nerdy.
Yeah, go ahead if you wanna talk now.
Yeah, I guess I had a few things that immediately sprang to mind. One is that I've just done the code for one of the steps that you've suggested, which was to calculate the climate for each river basin. I've written it up in R, and I'm happy to share. You just need the new boundaries and as input, and you could crank that out.
Oh yeah, I don't know how to do that yet.
Oh no, I've just
I'd love
done
to
it.
talk with you about it. I've done so far is generate the polygons.
Well, that's all you need, and the rest of it's done. Okay, that was one thing. The second is we are still struggling with we are interested in the big rivers, and we're interested in differences between the isotope values of the big rivers and the isotope values of the precipitation in their catchments, which is why I've just done that code that I was describing. Yeah. We're still having troubles with the precipitation, and particularly, I think, getting the high elevation precipitation isotope values and amounts right. And it strikes me that the high elevation small rivers could be really useful for correcting at least the isotope component of that precipitation model. And it may be that, we need to look at differences. That would be really good to be able to look at differences in our model between what we are predicting in the isotope values of precipitation at high elevations and either side of the Alps with what you're getting for those small rivers, and that could really help us.
Yeah, I think this would be a great project for a student to some extent, because we're limited to the road network, which actually doesn't get that high in most parts. That's part of the problem. Just going over the passes, you get a somewhat high, but I think really what you would need is, somebody to hike up into these higher catchments, right?
There's been some hiking done, Alice.
Yeah.
I was just gonna say, I mean, we have over 100 snow samples from various places in alpine locations. It might be an interesting exercise to compare the isotope values of those snow samples to your nearest headwater catchments to see how representative they are of those higher alpine areas. I guess the challenge here is that like snowpack, I mean, as you know, like the isotope values change as snow is susceptible to turbulent fluxes. And so I guess that's kind of the interesting question is, are rivers a good proxy to snow precipitation that we don't have very many in situ samples to help validate our models, or are they not because of all the fluxes that happen after deposition? I don't know, that's an outstanding question. In New Zealand, because it's so, it's so windy and you get so much melt freezing that happens, like I suspect that there could be a fair amount of variation after deposition. But that's kind of we collected all these snow samples and then we kind of had to ask ourselves, like, of what relevance is this from a hydrologic perspective? It's interesting. Like, I think we can use those snow samples as good values for understanding the precipitation values at the time of deposition, but that once it actually gets into the environment, it might look different.
Yeah, well, let's do it. Let's make that comparison. I can make a prediction, right, from our model and send it to you, and you can tell me how wrong I am. If everything works, then I think we have a really solid story.
Mm-hmm. Cool.
I really think that process of sending each other information and telling each other how wrong we are is how we will make these things better.
Yes.
In relating the precipitation predictions to the rivers and the river values to the precipitation, and then just inch our way forward.
Yes, I agree.
Sounds like another paper or something.
It sounds like a couple of papers, yeah, and also thinking about how to best design this moving forward. And anybody who's done peer-reviewed work before knows that the peer review process can often be painful, but you always learn something important from it. And I think this is an example of how we can recognize the limitations of what we did before and improve it on the next step. And that's why I'm happy to have you, Bruce, and you, Alice, involved in at least talking about this, so we can figure out best ways to move forward. It would be awesome if we could all get together sometime, maybe at a meeting or somewhere, to hash this out in more detail.
Are you still coming down for another winter sampling?
It should be 2 weeks from now. Yeah.
Well then, I mean, I'm here.
Okay, I'll get in touch.
Okay.
Once we have a schedule. So awesome. I think we should probably wrap up here.
Yeah.
Okay.
I might have to figure out where I need to kind of trim down the conversation. But I would like to thank you on behalf of the Environmental Research Institute.
All right. Thank you so much.
Yeah.